It is a race against evolution ultimately. The bugs are mutating, they are evolving, and they will get around any problem you throw at them. So then you have to consider both the risks and benefit of any intervention. So am I willing to give antibiotics to my child knowing full well the risks of giving the antibiotic? Antibiotics are not harmless. Yeah. They do have their side effects, some of them beneficial, many of them are adverse.
We have to have one world health. We need to look into the bat. There are not many mutations required by some viruses to make this hop as COVID did. So let's look at the bat now. Let's get the tests ready so that if and when the transition happens, we can see it arrive in humans. I will always know that it's bacterial or viral or both. So somebody gets sick, a strange viral illness, I shall know that it's viral.
This technology has to be affordable, it has to be accessible. The real test is whether this particular test is cheaper than a course of antibiotics.
Namaste, welcome to another edition of Sangam Talks Business. I have with me today Professor Andrew Shaw, CEO and founder of a company called Attomarker. This is based in UK, and he's bringing to the world and hopefully to India this new technology for early detection of diagnosis of disease. What that means is, and let me give a quick introduction, there is this thing called antimicrobial resistance. What that means is that bacteria, viruses, and so on and so forth become resistant to drugs. And I have some data here. About 4.95 million people in India in 2019 suffered from drug-resistant infections, and about 1.27 million died directly attributable to AMR, antimicrobial resistance. This is generally a huge problem in India, with off-the-counter disbursement of antibiotics, people going in and saying, I'm feeling cold, give me an antibiotic, and the chemist dispenses that medicine. So there's a lot of self-diagnosis that happens here. So India is one of the top countries in the world with AMR, and so it's a big problem here. What this technology could do is help separate what is viral infections and what is bacterial infections, for instance, and then be able to give the right diagnosis, right treatment, line of treatment to the disease. So Andrew, and I have Dr. Chandrasekhar, who's a former orthopedic surgeon but now part of Attomarker and is also linked with several investment opportunities around the world, especially in Singapore, UK, and you said, of course, India. So help me understand, what is the technology? Andrew, over to you first.
So the challenge is to work out whether you have an infection that is a bacterial or viral infection, or indeed both. It's possible to have both, especially with COVID at the moment. But if you give an antibiotic to a viral infection, it doesn't work. It has no effect at all. What it does do is affect all the bacteria in your gut. It will give you all sorts of side effects. And the more the bugs, the bacteria are exposed to the antibiotic, the more they learn how to get around it. And that's a problem. So what we need now is an accurate, rapid diagnosis that we can do anywhere, so that when somebody gets sick, as a parent, I'm worried about my children getting sick all the time, we have a period of time when you'd want to make that analysis. So the technology takes a pinprick of blood, we put it in a single-use chip, and the chip goes into something. This is sitting under an iPhone. And the iPhone is managing the test.
Where is the iPhone?
The iPhone screen, as you can see on the top, is embedded in the device.
Okay.
So it's interrogating the chip as it goes in, and now it's beginning to run the test. So your body responds rapidly to the difference between a bacterial and viral infection. We measure 3 things simultaneously, and as they begin to rise, either together or separately, we can tell you what sort of infection you have. If it turns out you have a bacterial infection, then it's right that I give you some antibiotics, and that will hopefully work. But the bugs are a bit smarter, so there are 12 types of antibiotics, and not all of the bugs work. All the antibiotics work on each bug. So also I could give you an antibiotic and it doesn't work. If it doesn't work, then I'll know it hasn't worked within 12 hours or so. We could retest, and then we could give you a different antibiotic. So what you don't want is your child sitting in hospital waiting to get better. The antibiotics you gave haven't worked, and you need to get the intervention right. What we need to do globally is reduce the use of antibiotics, so that we don't give them to the chickens, we don't give them to fish, we don't give them to the animals unless they need it, and us unless we need it. So smart diagnosis is what I want. 10 things from your blood in under a minute sometimes.
Thank you. How does this work? What is really the technology? What does the smart chip do? How does it detect viral infections from bacterial? If you could talk about all the non-proprietary things that you can share.
We have patents on the good stuff, so I can explain some of that. So it's a gold nanoparticle surface, and inside it there's an array that looks like this gold section here on the top.
Okay.
And we've invented this. This has come out of my research group at the university. It's very sensitive to the change in mass on the surface. It scatters light, and the camera sees the light change and converts that into a signal.
So the blood goes on this.
Blood goes on the top, and it flows down over the surface.
On all, okay.
And it and then the gold surface captures the molecules that I'm interested in.
Okay, how does the iPhone camera come into picture?
So the iPhone camera can see the chip, and it then says, oh, this is what it looks like now. When I flow the blood over the surface, it changes because I've captured what I want from the blood. And the iPhone says, oh, I can now calculate how much you've got. So it's simply by taking a picture of the array on the surface of the chip, the iPhone can do what it needs to do. So the clever thing then is the data from the iPhone go up into the cloud because they're all connected. And then we can start looking at the analysis of the result. We can make sure you measured it properly at home or in the rural parts of India. We could.
So basically, the blood has some sort of, I mean, finally a picture is a picture. But how do you know that this picture is a viral infection and this picture is a bacterial infection?
So we use the picture to measure three things. Something called C-reactive protein. Okay. It's a protein that everybody makes when they get inflammatory responses in their blood. We measure something called MXA, which is a marker that goes up when you get a viral infection. And we measure another one called PCT, which goes up also with bacteria. So imagine I measure a high concentration of CRP and PCT, but a low concentration of MXA, that gives you a bacterial infection. If I measure a low concentration of CRP and a high concentration of MXA, that gives you a viral infection.
Okay.
If all three are up, then you've got both, right? So it's a very easy decision for the doctor to look at and say, oh, this is a challenge, you've got some problem. Or indeed, I can get AI to look at that and say, yes, this looks like, I predict this is a bacterial infection with the following certainties.
Do you want to add anything?
So the traditional means of diagnosis of infections has relied upon identifying the bacteria or the virus and culturing them or seeing how they might respond to a variety of armaments we have, like antibiotics, antivirals. Whereas this technology relies on the host response, what our body, how it reacts to a viral infection or a bacterial infection, and these three biomarkers that we all produce and how they evolve over time. And because this is a near patient point of care test, and also because it can be done serially, we can see the natural history and how it evolves. So that gives us much more rich information on which we can base our treatment strategies.
And to be fair, doctors, if I present you with a baby or a child and presenting symptoms, you can't tell the difference between a bacterial or viral infection just by looking at the symptoms. So you need additional data, yes, additional diagnostics.
And also because there are a very finite number of symptoms, but there are 10 times, maybe even 100 times as many diseases. So we need a new science of diagnosis that is based on evidence, measurable criteria, so we can plot the symptoms against the diseases into which they might fit. And these kinds of hard evidence provides a specific guidance saying, okay, this could be this could be a different disease, and we know how it might evolve. So for example, infections, differential diagnosis of infections, whether bacterial, fungal, viral, or a mixed combination infection, that's one aspect. The other aspect, of course, is in a whole range of diseases such as metabolic disorders, allergic disorders, fertility hormonal disorders, neurodegenerative disorders.
So you can detect all of those.
This is a very powerful platform.
How? So there is C-reactive protein, MXA and PCT are three biomarkers.
For related to inflammation and infection.
But there are more.
Other biomarkers.
So on each chip you could have different biomarkers for liver, you could have different biomarkers for
each chip would have different biomarkers. Yes. I mean, so each chip would have be defined to say that this can detect only these three biomarkers. A different chip would be able to detect another set, or?
Yeah, another 7 biomarkers associated with liver function, for example.
Okay.
Or you could have 7 biomarkers associated with collecting dengue and TB and malaria infections. So When you choose the chip, the important bit at the beginning is there a first differential diagnosis? Is it bacterial, is it viral? I could then put another chip in and say, it's dengue. So if you have, the other thing is if you have the outbreak that's currently there in Bangalore of dengue fever, if the data are going to the cloud, I can tell you where I made the test. So you can see dengue is occurring in the northern part of the city, but yet hasn't spread down to the south yet. So in terms of preparedness for an epidemic or the next pandemic, the technology is going to be very well prepared.
So in effect, what happens is a collection of these devices, field deployable, they are affordable, and they're always connected. So they become a sentinel network. It could be infectious diseases, it could be in early stages of pandemic, or it could be, for example, just liver function in a cohort of patients who are undergoing, for example, treatment for tuberculosis. We have a massive burden of disease with tuberculosis still, and there is drug-resistant tuberculosis, there is drug-resistant leprosy. So in all these cases, we have a combination therapy for 6 months or 8 months, and then we have to test these people for any drug-induced liver injury, and that has to be repeated every couple of weeks. So instead of making the patients travel all the way to a laboratory every couple of weeks and then waiting for the results to come, these could be out in the field just with 1 drop of blood saying, okay, your liver functions are all right, so you continue with this regimen. But if liver function is getting damaged, then we switch to the other regimens. So these are monitoring sentinel networks.
And like you said, deployed at perhaps tehsil level or district
all over
level, wherever.
Yes. And we are aware of commercial and also public sector organizations that already are doing similar work out in the field, out in the rural areas. They are dealing at point of care, but this device is not yet available. So our mission is to make it available at India scale and at India price, so that we get as close to patient-centered diagnosis, patient-centered treatment, and in effect then it will be a wellness management. So that is the future.
There's always a challenge in diagnosis. You have to do a world-class Nobel Prize winning test for nothing because it has to be cheap, it has to be available to everybody. But I think with efficient manufacture here in India, we can make it work in Indian levels and Indian sizes.
And if it could work in India, possibly we can make it work anywhere.
And of course, I imagine the devices could be made much cheaper than manufacturing them in UK or...
Yes.
There are some real advantages. And I can take the iPhone off eventually and just put the camera in the box, and then any smartphone could talk to it. So we can really make it much more accessible. I think this could be in people's homes in time. Certainly you could look at your children, you could look at your elderly parents. Another cohort of people are people with dementia, for example, where describing symptoms is really hard. They get lots of urinary tract infections. We could pick that up early intervention, quality of life improves for these people.
And how many such biomarkers are there that you have access to or you have on your chips?
So it's
how many diseases?
It's an evolving field. So people like Roche have more than 260 biomarkers available.
So biomarkers are, how do normal tests happen?
So what happens at the moment is they take a big chunk of blood out of your arm, maybe 2 or 3 mils. It goes to the laboratory. The doctor will choose which tests to be done. And it goes into an instrument about the size of a huge washing machine, and those individual tests will be done with different methods of doing them. They've evolved in time. And what my technology does will unify all of those into the same way, in the same standard, mass standard.
They use biomarkers as well?
Yes, biomarkers are a field of differential diagnosis used by doctors. Been doing it for a long time. It's one of the few ways that doctors get an understanding of the chemistry going on in your body, because you can't see the molecules, right? So you have to make the tests. So there are some very well-known biomarkers. My strategy is to use the well-known ones, like for liver function, and then add 2 on the end that tell me something that's not readily available to give you something about liver damage. MXA is not readily available, but in collaboration, in conjunction with CRP, which is, and PCT, which nearly is, those 3 together are a completely novel collection, and that gives you new information.
So traditionally, diagnosis has relied on a couple of things. One is you take a blood test for biomarkers that are well known. The other, of course, is actually studying the organs themselves. For example, take a liver biopsy. Now, any intervention has its own risks and benefits. So you would not lightly consider doing a liver biopsy unless you think the information gained is going to be worth more than the risk of bleeding that is often associated. So for example, we can do biomarkers that are indicators of fibrosis of the liver, things like procollagen C3, C6. So if we can validate that prospectively, with the stated aim of comparing the results from a biomarker test with the actual liver biopsy, then it says this is as good, if not better, because it is less invasive. So this is how the protocols for the use of biomarker. And same thing with, for example, cancer diagnosis. There are tissue diagnosis is the ultimate confirmation, but there are a number of biomarkers that can serve as a proxy before biopsy. It does two things. For example, cancer cervix in India, and millions of women suffer because one, it's a modesty issue, they don't come forward. But if we can at least reduce the screening level, so you can identify patients at risk and then subject them to a more invasive procedure like a pap smear and things like that. So the idea of using these minimally invasive single drop of blood is to reduce the burden on the health service and make it easier for patients as well to come forward and have this done. And Andrew, perhaps you can explain a bit about the sandwich assay, because each biomarker has its own capture and detector molecule.
So the gold will sense everything in the blood. It will just change its color based on the change in the properties of the blood. But if I put something on the surface that captures the target, and then I put something on top that completes the sandwich, then it becomes specific entirely to the molecule that I'm looking for. If I take 10 of those simultaneously, I now get a 10-dimensional view about you.
Yes.
At the spot, right? It's not choosing one in the lab, it's not the doctor choosing which ones to do. 10 dimensions, fully standardized against you. So I now want to do generative AI and put in some 10-dimensional space and find out where you are. Is your liver shot already? Is your vegetarian diet good for you? Am I drinking too much wine? All these things, and what's the prognosis for my liver? How does it evolve in 10 dimensions? That, I think, could be the new diagnosis.
Yes, and often, traditionally, these diagnostic criteria express the results in terms of units. For example, insulin is expressed in units, liver function, the enzymes are expressed in units. These are functional units, and they have no, they do not indicate in any real sense how much of that substance is. Whereas with Attomarker, Atto meaning 10 to the minus 18 and based on a biomarker, it's important to understand that these are absolute mass units and standard against NIST units, so that we know there are so many attograms of this particular substance of a molecule of interest. Would that be a reasonable way of expressing that?
Yeah, one of my interests scientifically is putting together the models for how the body works. To do that properly, you've got to know how much stuff is there, how much get converted into other things, and how they move around. If those are not standardized properly, then you're guessing. But if I can do that and I can put the models together, then generative AI might begin to solve things like systems medicine. How's your immune system doing? How's your liver doing? How are all these systems collecting together to produce how we evolve in health and how we live?
And one of the, we could say beneficial, I mean, considering all the morbidity and mortality we had with the pandemic, with the COVID-19, one of the things we have begun to understand is the general effects of a viral infection in the post-viral sequelae or post-viral syndrome is associated with the number of viral infections, number of bacterial infections like Lyme disease and things like that. So your lab has done a lot of work on collecting the symptoms associated with the so-called long COVID syndrome, but being able to quantitatively measure the host response. What are the variants to COVID and what are the antibodies you've got? What is the quantity of the antibodies, and also what is the quality of the antibodies? So this generates a huge amount of data that then you analyze using AI, and you're able to identify different clusters of patients who either lack the ability to fight the virus or who do not generate enough antibody response. So based on that, then we can say, okay, maybe you need an additional dose of vaccine, or you need an additional dose of antiviral therapy, or an additional other doses of immunoglobulins. Would that be a reasonable way?
So the basic theory is that the virus hangs around. You don't clear it from your body properly. And I've now, I think, characterized the sterilizing serum. How does your body get over it properly, and how do some people not have a sterilizing serum? I think it goes for everything. So I think there's a chance of long dengue. People will not recover properly from dengue. There will always be some residual symptoms. There is long Lyme, all these other diseases are there. It feels to me like a new field. There's a type of response that's poor, and as Chandru says, we can now treat that or think about this new type treatment, endo-type treatment. That's really interesting. Can I change the way people respond to diseases to make sure we all get better properly?
Because we can show that there are gaps in our body's response to an infection. You can say, okay, these are the gaps, and perhaps we can fill that gap with these modalities, antivirals, antibodies, and an additional dose of vaccine perhaps. So this gives us an entry into treating other things like chronic fatigue syndrome. We really don't know what causes it. There are a number of hypotheses, but based on objective measurements of the immune response, the host response to these probable infections, we can begin to address these questions.
Very interesting. And you're here talking about the technology at other places. I believe you're talking at IIT Delhi tomorrow. Is
Yes,
that specifically on
long COVID and the persistence of the symptoms after the acute infection's gone? So yes, IIT Delhi and IIT Madrid later, Madras later in the trip. But it's a really exciting idea. The test is going to be submitted into a publication shortly, but the results look really promising, it's different. And it's only really possible because we've got this multi-dimensional view about what's going on.
And we were fortunate to be on a trade delegation from the UK. It was between the FICCI, Federation of Indian Chambers of Commerce and Industry, and the UK Research and Innovation and Innovative UK. So it was a competitive program, and Andrew secured a place for Attomarker. And there were 13 other companies who are addressing the problem antimicrobial resistance from a number of different angles. And the Attomarker instrument stood out as probably the most near patient methodology, a technology that can actually work very close to the patient and at multiple locations, and fastest technology as well.
But lots of smart people are thinking about antimicrobial resistance. Are there ways of keeping the rooms sterile?
Yes.
Are there ways of inventing new antimicrobials? Are there ways of putting things into open wounds so that they heal properly?
Yes.
All these areas, all these approaches to antimicrobial resistance have got to be used together so that the problem can be solved. The question is how to start. I mean, there are so many things that are going wrong. Do I invent new antimicrobials? If I invent them, do I shelve them so the bugs don't get to see them? Do I retire some antibiotics so that we don't use them for a while and then the bugs forget how to be resistant to it? All these things have to start somewhere. And for me, it's us, it's we, the consumer, saying, I want to know if my daughter has a viral infection or a bacterial infection and so really needs the antibiotic. For me, that sort of Gandhi would have appreciated, I hope, this civil action to change antimicrobial resistance.
Because it is a race against evolution ultimately. The bugs are mutating, they are evolving, and they will get around any problem you throw at them. So then you have to consider both the risks and benefit of any intervention. So am I willing to give antibiotics to my child knowing full well the risks of giving the antibiotic? Antibiotics are not harmless. They do have their side effects, some of them beneficial, many of them are adverse. So we have to strike a balance between what is good for the human beings. But equally, the other huge unaddressed problem in India, of course, is the animal industry, animal farming, cattle farming, chicken farming.
So you
poultry.
could do these tests for animals and
that is
birds as well?
That is the beauty. We are aiming at one health, one world, one health. So we treat the animal kingdom and the humans all as part of one unit. So these devices have been developed in collaboration with an animal
audience. You know, if the chicken that you eat is most likely pumped with antibiotics by default.
So the advantage I have is this small little drop of blood. If I could measure that from a chicken, which is possible, then you could make a random look into a wet market, for example, and say, this chicken is carrying H5N1, or this group of animals might be. The monkeys are very good at carrying viruses for us, so Ebola comes out of the monkeys nearly all the time. Could I take a little dart, grab a little bit of blood off the back of a monkey, and then do the analysis? So now I get a view about what the animals are doing. And that means things like zoonosis. This is a transfer of viral species from the animals to us. So we have to have one world health. We need to look into the bat. There are not many mutations required by some viruses to make this hop as COVID did. So let's look at the bat now. Let's get the tests ready so that if and when the transition happens, we can see it arrive in humans. I will always know whether it's bacterial or viral or both. So if somebody gets sick, a strange viral illness, I shall know that it's viral, but it would be really good to know whether it's something that come out of the bat and has made a mutation. So we're beginning to get to that point. I know Singapore is beginning to look at pandemic preparedness. WHO are beginning to think about it. So I think we should start sampling the animals and us to see how these processes are going to occur.
And to be really effective, then this technology has to be affordable, it has to be accessible, it could work at scale. And also ultimately the real test is whether this particular test is cheaper than a course of antibiotics.
Yeah.
So that's our hope, that we can make it work at that scale and that price.
Good. Thank you for bringing this technology out, and good luck to both of you. Incidentally, Andrew is in the process of fundraising at this point in time, and he's looking at setting up Attomarker in India separately with joint ventures with different medical device manufacturers, and there's probably some conversations already going on, as I understand.
That's true, yes.
So if you are interested, please approach Attomarker and Andrew, and for your investments. Thank you.
Thank you very much.
Thank you.